Welcome to Life in Biology. I'm Dr. Joel Graff, and this is episode number 16. Today we're going to be talking about adult stem cells. This is going to be a follow-up to episode 14 where we talked about pluripotent stem cells and embryonic stem cells. For adult stem cells, we're going to mostly be talking about hematopoietic stem cells because they basically serve as the paradigm for adult stem cells. Now, when you talk about stem cells, one of the first things you need to understand is that there are different levels of potency. So, totipotent cells are cells that can become all the cells of the body and all of the extra embryonic cells. And so these extra embryonic cells, you might think of things like the yolk sac and the placenta. So that's what totipotent cells can do, and this is these cells can occur when the sperm fertilizes the egg. You get a zygote, and the first several cell divisions of that zygote, the cells that come from that are totipotent. But if you allow several more cycles of cell division to occur, you end up with cells that start to become more and more differentiated, and they get to the point where they're only pluripotent. So in that case, they can only become the cells of the body. They can't become the placenta or vice versa. And so pluripotent stem cells, these are embryonic stem cells, and if you take these cells, you can end up with a full. If you take a human embryonic stem cell, theoretically you could end up with a whole human being from that cell. Multipotent stem cells mean that they can be means that your cells can become some but not all cells in the body. So this is a population of cells that self-renews, but some of the times when it divides, some of the cells can go off to further differentiate into other types of cells. And adult stem cells are multipotent, and we're going to be talking about an experiment where they one of the best first experiments where they're learning about adult stem cells, and the stem cell population was hematopoietic stem cells. So hematopoietic stem cells, are abbreviated HSC, are a paradigm for adult stem cells, which I've already covered. To study these cells, there were some interesting experiments that were done where you can take a mouse and give it a level of radiation that would be lethal normally, but the scientists would save the mice from dying by adding in cells from a non-irradiated mouse that could go in and replenish the hematopoietic compartment, which is your red blood cells and white blood cells. Now, to do this, what you would do is you'd collect bone marrow cells from a mouse that hadn't been irradiated and inject those into the tail vein of your lethally irradiated mouse. And as long as you used about 10 to the fourth and to 10 to the fifth, so 10,000 to 100,000 cells, you would be able to rescue the mouse. With 10 to the fourth, you only rescued it sometimes. 10 to the fifth, you rescued it pretty much all the time. And so they were able to estimate that about 1 in 50,000 mouse bone marrow cells is an adult hematopoietic stem cell. And then what they did is they wanted to take these stem cells. They knew it was kind of a mixed population of cells, and they wanted to try to narrow down the type of cell that is the hematopoietic stem cell. So the first idea they had was that if you're looking for the stem cell, maybe what you'd want to do is you'd want to get rid of all the cells that are expressing a marker of being a specific white blood cell lineage. So what I mean by that is that markers in this case are proteins that are expressed on the surface of a fully differentiated white blood cell, whether it's a B cell, T cell, macrophage, neutrophil, mast cell, eosinophil, basophil. These fully differentiated white blood cells can express specific proteins, and specific types of cells express specific types of proteins. So you take a mix of antibodies that can stick to these proteins that are on the surface of your differentiated cells, and you have those coating a plate, and then you put your bone marrow cells on that, and the bone marrow cells that have differentiated and become a little bit express these cells, these lineage marker cells, marker proteins, and they end up getting stuck to the antibody that's stuck to the plate. And so you just, from there, you collect the loose cells, and these cells presumably don't express any of the lineage markers. And so lin is short for lineage, so these are lineage negative cells. And by doing that, you get a pretty good enrichment. You go from about 1 in every 50,000 cells to 1 in every 10 to the 2. So that's 1 in every 100 cells. If you inject 100 lin negative cells into a mouse, you tend to rescue the mouse from dying after lethally irradiating it. And then a further step, it has been done where you select for cells that have been given the name LSK cells. And what this means is that they're lin negative, so they've gone through this process of getting rid of all the cells that express a lineage marker, but then they're positive for 2 different proteins called SCA1 and c-KIT. And so by having this combination, none of the lineage markers, SCA1 and c-KIT, about 10 cells with those phenotype can rescue a mouse from dying after being lethally irradiated. So they were able to narrow down the cell type that is the hematopoietic stem cell, and they've narrowed it down even further, and they have more defined factors. But the LSK cells are basically the famous characteristic of a hematopoietic stem cell. Now, they took several technical advances for this discovery or this characterization to be made. First was the assay where you were able to save a irradiated mouse from dying by supplementing it into bone marrow cells. But the second thing is the advent of monoclonal antibodies, and these are antibodies that are not a mix of antibodies. They're just many copies of the same antibody, and these antibodies can bind to a very defined target, and so that was helpful for these technical advances. And these antibodies were used for 2 different things. One, they were used for the panning. That's where you have the antibodies that the mix of antibodies that can bind to all the different lineage markers that are stuck onto the plate. So you use antibody for that portion of the technique, and once again that's called negative selection because you're selecting the cells that did not stick to the plate. And then the other breakthrough was flow cytometry, and the flow cytometry is an interesting assay where single cells get shuttled through a capillary one at a time, and there are sensors on the machine that can measure characteristics about that cell that's flying by. And for example, if you have the cells stained with antibodies against the SCA1 and the c-KIT, then you can tell the flow cytometer, hey, that's a population that I'm interested in. Are these double positive c-KIT SCA1 cells. And then a extra step of flow cytometry is fluorescence activated cell sorting, and this is where the machine can actually take the cells that have those characteristics that you want, put them in a separate tube than the rest of the cells. And this is called positive selection because you're physically, or the machine is using some sort of mechanism to grab those cells and get them diverted into a different test tube. So FACS is not facsimile, it's fluorescence activated cell sorting. So from these experiments,